Hello and welcome to this class on biostatistics. Let us understand the meaning of the term biostatistics. Biostatistics is nothing but application of techniques of statistics in biological sciences. Essentially, biostatistics is nowhere different from the statistics, which is a branch of mathematics. The only apparent difference is in biostatistics, we are only considering the examples of biological sciences. And what do we do in statistics or biostatistics? We collect the data, we organize the data, we summarize the data, and we analyze the data in order to make predictions. I'll give a very simple example. Let us say that you have about 100 acres of land and you want to predict what will be the total yield of crop at the end of the season. You need not cut the whole crop to know the outcome of your crop. What you can do is you can only take some samples here and there, and you can estimate the amount of crop produced by the samples and multiply it by the total area, so that you'll get the total expected yield of the crop of that area. So this is in simple terms what is statistics. In statistics, there are two things that are very important. One is population, the other one is sample. Population is the entire body of the data, entire body of the experiment, or entire body of the crop, or entire body of whatever you have considered for research. For example, let us say you want to know the blood group of a person. Alright, each and every person has about 5.5 liters of blood in his body, 5 to 5.5 liters of blood in his body. If you want to know the blood group, you need not draw out all the blood. What you can do is you can take out only a few drops as a sample and use it for the estimation. And only by estimating a few drops, only by finding out the blood group in those few drops of blood, you can tell the blood group of the person. So here the entire blood of a person is called population, and few drops of blood that you considered for study is called sample. If you look into the history of statistics, the first name that comes to my mind is Rituparna. Rituparna was a king living about a few thousand years back in India. He was asked to count the total number of leaves in a huge tree by his guru. What he did was he selected some branches, they are supposed to be the samples, counted the number of all the leaves in all the branches that he collected, and took the total, and the total was divided by the total number of branches that he collected, and he got the average number of leaves present in a single branch and counted the total number of branches in the tree, multiplied that value with the total number of branches to get the total number of leaves that are present in the tree. So this is one very strong evidence to say that an Indian king called Rituparna is one who used statistics in biological sciences or in sciences for the first time. As far as the European biostatisticians or statisticians are concerned, we have two names. One is John Grant, the other one is William Petty. Both these are British scientists who used statistics for demographic studies. In fact, William Petty made a simple survey of births and deaths in London, and for the first time where he used statistical methods, and that is how the branch of statistics came into being and started developing. There's one name from India who is known worldwide as a big statistician, as a great statistician, that is Prashanta Chandra Mahalanobis. There are three big names that have contributed enormously to statistics. They are Carl Pearson, Francis Galton, and Ronald Fisher. As far as steps or stages in statistics are concerned, in the first stage what we do is we collect the data, summarize the data, and graphically represent the data by using histograms, bar diagrams and things like that. In the second step, we start analyzing the data. There are two ways of analysis. One is to find the central tendency. It is like finding the mean, finding the average. So we have measures of central tendency where we calculate mean, median and mode. The second step in statistics is finding the values of dispersion, measures of dispersion. It is to give importance to the data that is distributed around the central value, and this is calculated by range. This is calculated by calculating or estimating the standard deviation and variance. In the third step, what we do is we go for hypothesis testing and estimation of ANOVA. ANOVA is nothing but analysis of variance. And the last stage where we make predictions based on our analysis. Another important thing that we can learn from statistics is correlation and regression. Correlation is a concept where we can understand the relationship between two different parameters. For example, we are studying the two different parameters like height and weight. We can estimate the relationship between height and weight. As we all know, as there is increase in height, there is increase in weight, usually. So this is what is correlation. What is regression? Regression is one where we can understand, or you can come to know about one factor with the help of the other factor. For example, if you know the height, you can know the weight. Similarly, if you know the weight, you can know the height. One more example is you can find out the correlation between temperature and light intensity. These two are related. Where there is more intensity, there is more temperature. So in a nutshell, in statistics, what we do is we collect the data, we summarize the data, we graphically represent the data, we measure the central tendency, then we measure the dispersion, then we estimate the ANOVA, that is, we analyze the variance, and finally what we do is we make predictions. Predictions are ultimately the outcomes of statistical analysis. That is it about statistics in this part. We will learn more about statistics in the next part. Thank you for watching this presentation on statistics. If you have any queries, any doubts, please always contact me. Like, share and subscribe.